Good morning students. Today we are going to discuss few bacteriological tools on exam perspective. So this is a universal container. This is used for urine collection as well as for sputum collection. Okay. You can also have plastic containers also. So this is a screw cap container. Okay. Fine. Next. This is Bejos bottle. This one you use for preparing media like low flush serum stand. This is a stool sample collector. Okay. As I told you, this is also an universal container which we use for sputum collection. This is a cotton swab, a petri dish used to prepare media. This is a loop, bacteriological loop used for streaking or inoculation. This is a Bunsen burner. This is an anaerobic jar. This is used for anaerobic culture purposes. Fine. This is an incubator. So I think everyone knows what is the use of an incubator, right? Fine, need not explain. Next is hot air oven. What is the temperature of hot air oven? Very good, Dr. John. 160 degrees Celsius. Okay, fine. It's used for sterilization. So you should know what are the instruments that you should use for sterilization in hot air oven. Fine. This is an autoclave. Now we'll come to culture media. So first we'll talk about bacteriological media. The use of this media is to isolate the organism from clinical specimen, then culturing the bacteria, which is an initial step in studying the morphological and identification. Okay, so the main purpose of this media is to isolate the bacterial pathogens from samples, that is the clinical samples, then culture it, identify, then go for antibiotic susceptibility testing and so on. Fine. So what are the contents? So hence an artificial media, we should provide all the nutrient components that are necessary for the bacterium to grow. So basically culture media contains water, a source of carbon, a source of nitrogen, and some trace elements which are required for growth and other growth factors necessary for the bacterial growth. Other than this, the pH should be optimum. The oxidation tension, osmolarity should also be taken under consideration. So media can be classified based upon the properties. The first one is physical state, the chemical composition, and the functional type. Based upon the physical state, they may be classified into liquid, semi-solid, and solid media. So first we'll discuss about liquid media. So liquid media are water-based solutions which are generally termed as broths, milks, and infusion. Liquid media are available for use in test tubes, bottles, or flasks. In liquid medium, bacteria grow uniformly, producing general turbidity. So turbidity is an indicator of bacterial growth. Liquid media tend to be used in a large number of bacteria have to be grown. Okay. Next, liquid media are suitable to grow bacteria when the number is in the inocular. So these are few examples. Okay, semi-solid. So this is prepared by reducing the agar concentration by 0.2 to 0.5%, which renders a semi-solid concentration. So such media are fairly soft and useful in demonstrating bacterial motility. Certain media such as Struart's and Amy's media are semi-solid in consistency. Okay. Oxidation fermentation test media is also an example. So these are all the examples of media, okay, which are semi-solid in nature. Next is solid media. Any lipid media can be made into solid media by adding a solidifying agent, which is usually an agar. So it melts at 95 degrees Celsius and solidifies at 42 degrees Celsius. So agar does not give any contribution for nutrient property, and it is not hydrolyzed by any bacteria and usually free of growth promoting or retarding factors. So solid media contains around 1 to 5% of agar, which enables the formation of district colonies, and the media is reversible to liquid on heating. So these are some examples. Next, the chemical content. So here we can divide them into two main categories: the synthetic media and the non-synthetic complex media. Okay, so synthetic media contains pure organic and inorganic compounds that are chemically defined. Fine, these are the examples. So this table will show or give the examples of medium of growth and maintenance of green alga euglena. So this is just an example. Fine. So this is a complex enriched media containing ingredients that are not chemically defined. That is, they are animal extracts. This is a blood agar. Fine. So based upon the type of growth, media can be collected into simple, selective, enrichment, differential, and miscellaneous. So a simple media, for example, you can say peptone water, nutrient agar, which can support non-fastidious organisms. Simple media are not those media which contain minimum nutrients which are possible for colony growth. So they contain only basic materials which are required for colony growth. Okay. So simple media contains a carbon source, for example glucose, various salts which may vary among the bacterial species, and the growing conditions which are generally provide essential elements such as magnesium, nitrogen, phosphorus, and sulfur, which led the bacteria to synthesize protein and nucleic acid. And the most important content is water. Selective and enrichment media. So these are designed to inhibit unwanted commensals and or contaminating bacteria and help to recover pathogens from a mixture of bacteria. Both these media serve for the same purpose. Various approaches are made to make this medium a selective one by the addition of antibiotics, dyes, chemicals, alternation in pH, and a combination of these. So any combination of these will make the bacteria selective. Okay, so it means that if you want to grow specific species of bacteria, you make the media specific for that bacteria. Okay, by using such media you can inhibit the growth of the commensal organisms, making it a pure culture. So example is Thayer-Martin media, which is used to recover Neisseria gonorrhoeae as well as Neisseria meningitidis, which contains antibiotics like vancomycin, colistin, and nystatin, which are gram-positive bacteria specific. Blood agar and chocolate agar are also examples. Next is enrichment media. Enrichment media are liquid media that also serves to inhibit the commensals in the clinical specimen. So other examples are salinity tetrathionate broth and alkaline peptone water, which can be used to recover pathogens from fecal specimens. Differential media. So these media are designed in such a way that they differentiate bacteria based upon certain properties. For example, McConkey agar will differentiate them based upon lactose fermenting colonies as well as non-lactose fermenting colonies. So various approach includes incorporation of dyes, metabolic substances, and those bacteria that utilize them appear differently colored colonies. So as I told you, McConkey agar is an example. Other examples are CLED, TCBS, and XLD. So selective media enables one type of bacteria to grow, while differential media allows bacteria to show different reactions. I think you understood the concept. Fine, we'll go to the next slide. So examples of miscellaneous bacteria are reducing, fermenting, and transportation media. And examples are, what is this in the left? John, answer. Why are you keeping quiet? This is TSI. Okay, fine. John, you should go home and study well. Nowadays your preparation is not good. Okay. Okay. Next. Transport media. These are used for temporary storage of specimens which are being transported to the laboratory for cultivation. Such media are ideally maintain the viability of all organisms in the specimen without altering their concentration. Transport media typically contain only buffers and salt. The lack of carbon, nitrogen, and oxygen growth factors prevent the microbial multiplication. So here the number of bacteria are stable. Okay, they don't multiply, but they are viable. For example, you can talk about thioglycolate broth, which is used for strict anaerobes. Okay, and also Schwartz medium. Schwartz medium is an example for, it's a homework. Fine, you will type in the comment section.